Simple Staining Lab involves the use of simple stains, which are water-soluble and alcohol-soluble, sometimes lipid-soluble stains that stain bacteria only one color. For example, methylene blue stains bacteria the color blue. They are mainly basic stains with a positive charge, and because bacteria have DNA that's negatively charged and they have slightly a negative charge in their cytoplasm, they are attracted to these simple stains. Simple stains inform us only about the bacterial shape and the arrangements. Let's take a look. Here we have a coccus shape, bacillus shape, and a vibrio shape. Here we have a coccobacillus, spirillum, and spirochete shape. If we arrange these together, we might have an individual coccus, no real arrangement, we call that single coccus, or two cocci together, diplococci, four cocci together, tetrad, a chain of cocci together, streptococci, or a cluster of cocci together called staphylococci arrangement. Couple more arrangements. A single bacillus is called a single bacillus arrangement. A chain of rods, streptobacilli. Let's move on to the materials. We have the baccinerator and our loops, the distilled water. We will use some stains. Here's crystal violet, a violet color. Here's carbolfuchsin, a deep red color. And here's methylene blue, blue color. They're all simple stains. We also will use the regular microscope slides that you can see here. We will also need cover slips to cover our slides, the little tiny square pieces that go on top of the specimen to protect the lens from the stain and the bacteria. Further, moving to the more materials, we have the immersion oil, which we will use with our 100X. To clean our immersion oil, we'll need the lens wipes and the cleaner, making sure we get all the oil off the lens. And we will also use bibulous paper to dry our slides. Some instructors may use a disposable loop. In this video, we will use the metal loop. If you use a disposable loop, just make sure you put it in the hazard trash. Let's get started. Here's our staining rack. You can see some slides drawing on the staining rack. You'll notice also that there's a Tupperware under the staining rack to collect all our stains and our waste during the staining procedure. We have a microscope back here so we can view our specimens after we stain them. Moving on to the bacteria that are used in this lab, we have E. coli in a liquid medium, Staphylococcus aureus in a solid slant. I don't know if you can see, there's some film of bacteria growing on the slant. So this is Staphylococcus aureus. And we also will use bacillus. You can see a film there growing on the slant. Some instructors may prefer liquid bacteria, bacillus, actually liquid for all of them. Labeling our slides. The first thing we'll do is we'll label 3 slides, 1 E. coli, 1 Staph aureus, and 1 Bacillus cereus. Let's look at our slides. We have labeled bacillus on the far left side. We don't want to label in the middle because the ink will interfere not only with our staining procedure, but also with your microscopic view. So there's our bacillus slide labeled. Here's our staph slide, again labeled to the left side. And here's our E. coli slide. We have loops that we have sterilized and cooled. We pick up our E. coli and now we will create smears on slides. We dip our loop into the E. coli culture. We go to the bottom, get a good sample. We don't want to do too concentrated smears because then they will be too thick to stain, and we also don't want cultures that are too old because the staining won't work. So here we're creating a smear on the slide for E. coli, dime shaped to quarter sized. We want to spread the bacterial cells nicely on that slide. We sterilize our loop. And now we will move on to our Staphylococcus aureus slant. Now notice this is going to be very concentrated because it's more slimy and there's a more concentrated look to the specimen. So we're going to put a drop of water, distilled water, on each one of the other slides, the bacillus and the staph, because we're going to be using the bacteria growing on slants rather than on liquids. We pick up our staph with a clean, sterilized loop. We go in and gently scrape off the surface, not the agar, but the actual bacteria. I don't know if you can see that. There's some bacteria that are kind of liquidy there. This is staph off the surface of the slant. Again, we don't want to pick up too much of a sample, just enough to go and smear in the drop of water that we placed. We create a quarter-size smear, as you can see here. We will sterilize our loop. And now we will create our smear for bacillus on the bacillus slide. Here's bacillus. You can see the film growing on the slant. We will use our ring finger and our pinky to prevent the cap from touching anything and getting contaminated. We will go in and barely scrape off some of the slime, the bacterial slime, off of the surface of the slant. We will place our specimen into the drop of water and smear, trying to spread the cells as much as we can into a quarter-sized smear. And this is for our Bacillus cereus. We will now sterilize our loop, and we have created 3 smears. So after labeling and creating the smears, we will move into the fixation step, which means we adhere the specimen to the slide. We do this in 2 ways. First, we air dry and then we heat fix. We will wait now to air dry these slides for about 10-15 minutes. 10-15 minutes are over. We have clipped our slides. You can see the smears here have air dried. And remember, we have to fix them, glue them to the slide. So we have air dried them for about 10-15 minutes. They are very nice smears, ready to heat fix. And here's our 3rd smear. You can see they're nice dry smears. And now we will heat fix them. All we do is pass the slide over the flame for 2-3 times so we can make sure that the specimen really adheres to the slide. We don't do the heat fixation first before air drying because we can kill the bacteria and then the bacteria will not be able to adhere to the slide or be stained. We will put away our flame, and now we will move into view our nice staining rack. We place the slides and unclip them onto the staining rack. And now we are ready to begin our staining procedure. Your book has the protocol for this simple staining procedure. You will notice we stain for about 2 minutes and then we wash with water and blot dry our slides and put them under the microscope. We have chosen carbofluxin for the E. coli, so we will flood the E. coli smear for about 1 to 2 minutes with carbofluxin. We will choose methylene blue for the Staph aureus specimen. Again, we will flood the specimen smear with methylene blue. And now we will use crystal violet for our Bacillus cereus sample. We will wait 2 minutes. We'll get a close-up here. Our slides will be just sitting there for about 2 minutes. The 2 minutes were ready. We make sure we cover the label with our finger so we don't wash off our labels. Then we won't know which bacterium we're looking at. We take our distilled water and spray water down the slide facing the Tupperware, trying to get as much stain off that is not attached to the cell, the bacterial cells. We wash in the front, and sometimes we will have to wash in the back as well of the slide. But you can see this slide washed very well. The specimen is still on there, the smear, so that's good. Again, we cover the label and we spray the water, let it run down the slide, clean the excess stain. Here we clean it on the back, and you can see it's nice and clean. We check our smear, and our smear is still on there, and it is blue from the methylene blue, so that's great. Our third slide, same procedure. And now we have completed washing all 3 slides. The next step would be to dry these slides. We pick up our bibless paper. It's like a little booklet. We find 2 pages so we can put our slides in between in order to dry them. Just like that. We close our little booklet and we pat dry. Most of the times the slides don't dry with the first attempt, so we will find another 2-page area where we can put our slides in between 2 pages of our bibless paper and dry them once again. Our slides now are ready to view under the microscope, but remember, we don't want to get the stain or the bacteria on our lenses. Our smears look very nice. They're all nice and dry and stained. So we do not want to get stain onto our lens or bacteria, so we will be using a cover slip for each slide. All you have to do is place the cover slip on top of the specimen. You can see the cover slip, it's a little square piece, and move it so it covers your whole specimen. And now we are ready for the microscope. Using the microscope to view our cell slides, we will pick up the E. coli slide first. Make sure we clip it. We can move it, the mechanical controls, north, south, east, west. We can use those to move the mechanical stage. We want to center our specimen so we can see it where the light comes through, and we set it at 4x. We look into the oculars, and using the coarse knob, that thick knob, we bring the stage up, 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 until we find the specimen. Once we see the specimen, we see the bacterial cells, and we can actually determine the shape and the size and the arrangement and all of that, we stop at 4x. We'd like to go higher, we switch to 10x magnification. We don't touch anything else besides the fine knob, and you can see here we're adjusting the fine knob until we find the specimen, and we have found it. Now we switch to 40x. Do the same thing, adjust the fine knob until you find your specimen. Once you find the specimen, you want to switch to the immersion oil 100x view. And the way to do this is that make sure you have everything in view at 40x, switch to a V. We will put immersion oil in between, right where that little light dot comes through the condenser. Be careful not to switch the 40x, but the 100x into the oil. You don't want oil on the other lenses. Then you have to clean them. We've switched the 100x. We play around with the fine focus knob. Notice here I am adjusting the light using the iris diaphragm located on the condenser just to get a better, more clear, crisp view of the bacterial cells. This is an iPhone that is connected to a device that can go through the ocular piece, and it's showing you here Bacillus cereus under the microscope, 100x immersion oil, and I'll make that a little bigger and you can actually see the bacilli. And they are purple because we stained them with crystal violet. All three slides will be viewed under 100x to determine the shape and arrangement. Please remember to clean your immersion oil, your area, and put away your microscope. Photos will be provided to students for the completion of the lab report. Thank you.